Ja.
So I want to welcome you all to this first session of this great Congress. I'm Åsa Petersén. I'm a professor and psychiatrist at Lund University in Sweden. I have the honor to share this first session with Filipa, Júlio, who's a board member of EHA. In this first session, we want to set the stage for the whole conference by inviting in experts from different aspects of Huntington's disease, from an HD family, from an HD clinic, and from a laboratory that's looking at the real molecular aspects of Huntington's disease. And so we're going to begin with the most important part, the HD family.
Yeah. So hi everyone, it's really a pleasure to be here. And I'm delighted that I'm able to present Anne-Elisabeth to all of you. Anne-Elisabeth has a very diverse background, having lived both in Colombia and Sweden, which has given her a unique perspective on the global impact of Huntington's disease. In the HD community, she has multiple roles. Her experiences as a person at risk and a former young caregiver have fueled her passion for advocacy and engagement, especially among young people. She's currently involved with the Swedish HD Association and also with HDYO and EHA, and more recently, she has become actively involved with the HD community in Latin America, particularly in Colombia, where her mother is from, and where she has been studying for the past 5 years. As an HD advocate, she is dedicated to foster the involvement of young people with the community, bringing them closer to the associations, clinicians, and researchers. Anne-Elisabeth believes that by working together, we can navigate these times of realistic hope and drive meaningful progress in the fight against Huntington's disease. So let's hear the voice of the young HD family members and give a warm welcome to our special speaker, Anne-Elisabeth Magnusson.
Which one am I talking to? On this one? This one? Okay. Well, good morning. My name is, as you can see, a very long one, but my name is Anne-Elisabeth Saldarriaga Velez from my mom's side and Magnusson from my Swedish dad. Today I'm talking about experiences from HD family and how important it is to be able to work with clinicians and people from the laboratory in general. I was a former caregiver. I was born with an uncle who already were in the mid stages of HD, and afterwards at age 5, my dad started having symptoms. And he got diagnosed when I was 8 years old, and around that age was the time I was a caregiver. So I've been a caregiver since I was around 6 years old until I was around 23. So I'm currently at risk. I'm the only one left from my family side, on my dad's side, that have maybe a chance of having Huntington's disease, or as we say in the community, or not, because we have to look on both sides. And I'm also, as Filipa said, a representative. I work for the Swedish Association. I work for the HDO as an ambassador. I work together in HDCAP, the European Huntington's Disease Network, as well as the European Huntington's Disease Association. So, and there is also one right now this year that is the Colombia one. So I'm very involved, and that's what I'm going to continue to talk about. So let's see here. So a formal introduction, I think it's very important that you know how families might look like. So this is a picture of me when I was getting baptized. My mom on this side is the Colombian one, and then my father, who is Swedish and Norwegian, and where the gene came from that we know were tested was my uncle, and my old grandfather, who we know had HD as well, but sadly he passed away around when my dad was 18, so I didn't have an opportunity to get to know him, but we had pictures. So, understanding Huntington's disease, how does it impact families and friends is very crucial because it profoundly impacts us all. It's not just about the patient, as we have mentioned before. It's something that brings emotional, physical, and financial challenges. As family members such as I and many people become caregivers and having to face uncertainty about their own genetic status and cope with things that is like the gradual loss of someone with HD and their abilities and the burden of having HD in the family for maybe generations and how it affects us. It's also important to know that it's not just the patient, it's family, and then we have friends as well. And it's important that we try to keep these relationships, and we have to make sure that everyone gets what they need in order to make sure that the patient has the resources, and when the patient has the resources, the family has the resources, the friends has the resources, it becomes a very important connection where we all can help each other and make the situation easier, especially when it comes to mental health for everyone, basically. My family story, where, like I said, it started when my dad, when I was around 5 years old. My uncle had it when I was born. He got it in 95. I was born in 97. And my dad was the caregiver, but then in the middle of it all, I started getting involved with my uncle. I really loved him a lot. I was probably that kind of child that I didn't care about, like if you had involuntary movements, or I was in a special kindergarten where all my friends had different diagnoses, but I didn't see them as diagnoses. I just saw an opportunity to get to know a person. And I think that's something to be really aware of, that it's not just HD, a diagnosis. It's about the person. It's about the family behind it all. So that was something I learned from then. And because the caregiving situation on my dad's side was difficult, since my father already lost his father when he was 18, 20 years old. And then the brother got sick, and then my dad got sick. My dad obviously got some really complicated depression. And that made it so much harder for everyone in my family to handle HD. And in that case, as a 6-8-year-old child, I thought it was my fault because I noticed something was wrong with my dad, but nobody would tell me why. So I started like helping my dad with small tasks, and then that escalated to something bigger, such as taking care of his economy, going to go grocery shopping at age 8, basically being an adult since a young age. And nobody really, during that time that I was a caregiver, the adults didn't even care. They thought I was a good child, that I was an angel, that I was a miracle happened to my dad. But nobody, when I asked for help, when my mother asked for help, nobody could help us. And it was really, really complicated with the caregiving situation, that if you're alone and it's a rare disease, you don't have everything that you need to take care of a person, really. So as I grew older, that's when I got involved with association as 10 years old, and then I started going to HD camps, HDO camps, and that's when I started getting the help I needed and the right support, the right information. And with that, I could finally become more involved internationally to help others in my situation together with these associations around the globe. So, for instance, with the AHA, Moving Forward umbrella, we have the testimony on the impact of the 30th anniversary of the gene discovery. It was something I was involved with because that's where I started sharing personal and family experiences, as well on how important it is to have had this opportunity to know the gene discovery, because this helped with emotional, psychological, and practical challenges faced by not only my family, but other families navigating life with Huntington's disease. We had surveys for young people where we translated them and made sure that they came out to the right people. And with that, we designed things in how we could make sure that the needs, challenges, and experiences of the youth affected by HD were highlighted and how they could get the help they deserve. And also, like as to say, make sure that all of these voices are heard, because it's not just me, I'm not the only one standing here. On my shoulders, I have all my friends and family members from the HD community with me today. I even have my father here in this necklace, so I'm here representing them all together. So we're doing that. And when it comes to turning these challenges into advocacy, it's very important to do so, because by being an HDO ambassador, by being an HDCAB member, by being a volunteer work, both when it comes to the Swedish Association, but now that I've been able to come to Colombia, live in Colombia, and know my mom's side, looking how Latin America works compared to Europe, that's when you discover how important it is to have the ability to do some kind of advocacy and make sure that all of these voices are heard, because there are so many people that need help right now and that are invisible, especially in Latin America right now. I wasn't sure how the situation were, but 2-3 years ago when I got involved, you start noticing how severe the situation is compared to Europe. And still here in Europe, we still have issues with, for instance, Sweden is still not able to be in Enroll, so you can't register in Enroll, which is a big negative step that has been going on, that we've been fighting, because we need all of these things to make sure that people are being visible, heard. And that's why, for instance, being an HDO ambassador, I have the opportunity to work together with other youths and being able to help young people around the globe who doesn't have the help that they actually need. Same with HDCAB. Being an HDCAB member is super crucial, because that's when we, as family members, have an opportunity to work on the clinical side. So it's not just them talking. We have a family behind everything that explains how the disease works in reality, and as well on how it looks on the family side and our own view on everything. And the same with by volunteering is super important, because by doing these volunteer works, you start noticing how maybe we're a small community, but in reality, when we start having a cooperation between countries, between other members, this rare disease is not so rare anymore. We start growing, and look in this room, it's like a huge amount of people that's being here. So it's actually a great opportunity working with other people, and I really recommend that you get to know each other to continue that. So that's something, looking forward is something that is my main goal in everything, expanding these HD associations, because people need these supports, especially underserved regions, to provide localized support, drive advocacy, and promote research everywhere. especially improving technology. Collaborative research is really important as well, because having these promoting international cooperations in HD research, we can foster new leaders who will bring fresh perspectives and continue to drive progress in HD advocacy. So it's really important to value young people. And together we can build a global network that provides comprehensive support and moves us closer to finding a cure for HD. And if it's not a cure, at least with treatments, that is equally important, because we have noticed that compared to 20 years ago, that's a big step with what's going on right now. I've seen improvements myself, being a caregiver, but also working at a health home for HD patients, and seeing everyone getting the treatment and the help and having this important network with their families and their friends, like we told before. You see improvement, so it's very important that we continue this way. So I thank you all for your attention. I hope you have a good couple of days. Please do continue to work with each other, and if you meet new people, making sure that you really get new networks as well and know the true value of working together, because being a global advocacy is so important, because then we can help people that doesn't have the resources and doesn't have the opportunity to be here, to talk here. So yeah, that's what I have presented today, together with all of my friends and ambassadors.
Thank you so much, Anne-Elizabeth, for all these nice thoughts and sharing all this.
And now I have the honor to invite Professor Alexandra Durr to come up to us at the podium. Alexandra Durr, she's one of the leading clinician scientists in the HD field. She's a professor of medical genetics and a senior consultant in neurology at the Paris Brain Institute. She has received numerous awards for all her work for HD, and she has pioneered the first pre-symptomatic genetic testing for HD in France, and since then has followed HD families. We're very much looking forward to hear Alexandra share her experiences with us today.
Thank you very much for inviting me and for letting me talk about some experience from clinics. And I would like to thank Anne-Elizabeth, because I thought it was the perfect introduction to start thinking what can we do. And I have the pleasure and really the honor to have a team that is clinical and doing research. So not only the EHDN has 20 years of anniversary, but the neurogenetics has now 50 years of existence and have a history now. So one of the fathers of this neurogenetic history is Tom Bird. And I just met him in Seattle, because we were celebrating the 50 years of neurogenetics in the Washington University there. And interestingly, when I talked to him about all the advances in neurogenetics in general, he told me that the only thing he was really eager to do at the end of this experience is to write a book about the turbulent world of Huntington's disease, with the nice title, Can You Help Me? And so I thought that the neurogenetic knowledge, his knowledge and our knowledge, should really be used in the clinic to answer questions of all the families, of the people who we're meeting and we try to answer. Someone to give answers, and sometimes to give things that we don't know, say we don't know. And so I thought I would choose 3 questions that I would like to answer with you in the minutes I have. Am I and my family at risk for Huntington's disease? And if so, how much and what can I do? And if I'm a carrier, how do I know I have the disease? What are the determinants of the age at onset or of the onset of the disease? And since this is not so straightforward, as you're going to see. The third question would be, what is going on during the silent phase before the onset of the disease? So I will take these 3 questions, and the first one, am I at risk? And so what we do is, my parent has Huntington's disease, what is my risk of getting the same? And what we do in clinics is we draw a family tree, and we look if the person here with the arrow, can I do this? Yes. With the arrow is at 50%, or if we are talking about his girl who is at 25%, and we talk about the father and the family, and as Anne-Elisabeth said, about the whole thing around. And then the question comes up, but 50% at risk, okay, but how come 50? And 89% in my clinical experience say it's because I have a mother and a father. And at the end, it's not really true, because what you can see here is that this person who has Huntington's disease has a normal allele, which is polymorphic, sure, and has the expanded allele, and there the 50% is coming from. And this is somehow reconciling with the parent who can transmit the disease because he has also the normal allele he can give. And Anne-Elisabeth said, getting it or not getting it, that's the question. What you can see here too is that the normal allele is quite monoclonal, and you can see in this fragment analysis that the expanded allele looks different. And this is the somatic instability. We come back again because these are many questions about this too. So the next question comes then often, but if I want to know if I'm at risk, if I'm a carrier or not, how many at-risk persons ask for that genetic information at the end? And we all know, 30 years after the identification of the gene, that there is still a very low uptake in all countries throughout the world, ranging from 5 to 25%, who are people opting for the test. And the numbers here you can see from the, no, you cannot anymore because I changed the thing. The numbers you can see here, Huntington's disease is in our clinic the most often disease where people ask for a genetic test. Perhaps it's more prevalent, but if you look at the ALS community, for example, with the C9orf72, who was only identified in 2011, probably it will, because it's a more frequent disease, have more people asking for the test in 20 years to come. The next question is then, but why is there a low uptake like this? And we did with my colleague Marcela Gargiulo, the psychologist in my team, and Lucy Pierron, geneticist in my team, we did 2 questionnaires in 2000 and 2020, asking people who should inform and how, at what age you were aware, and what are the motivations to be tested, because we thought we had the hypothesis that in 20 years of time, things changed. And you can see here that the age at HD awareness is quite late. Again, Anne-Elisabeth set the stage. She said at 40 she understood things that she didn't understood when she was 6 and her father had the disease. And it's true that it's around 30 years old that people understand that they're at risk. And the same thing is for the motivation to take the test. Between 2000 and 2019, there is not much difference, because it's still the right to know that prevails, and even more now in 2019, or in now, because I thought it would be more the benefit from treatment, but it's not there yet. The second question that comes up a lot is, how do I know when I'm a carrier I have the disease? How does it start? Who knows? You know, I know, my father, my mother, my friends. Who's going to notice what I have, and what is determining the age at onset? I quote Tom Bird again from his book. I hope you had the chance to read it, because he says the variability, the span of age at onset in this disease is nothing like, you don't find this in Alzheimer's, you don't find this in Parkinson's disease, and you don't find this in schizophrenia. This age at onset range, it's enormous. It starts at 5 and ends at 91, I think. So it determines what we know today is very clearly the size of the expanded repeat, and I come back to this, and the sequence of the expanded allele, but also genetic modifiers They explain this very large variability here, but in this Manhattan plot from the meta-analysis of genome-wide search for frequent variants, there are significant peaks to explain what is not explained by the CAG repeat, but it explains only about 2%. So there are other genetic players that we have to identify probably. And one other player in the age at onset or in the disease driver is probably the somatic instability of the repeat in the neurons, because it can expand into hundreds of CAG repeats in a neuron during lifetime. So again, it was very important, and it's important to use this in the clinic, that the repeated allele is correlated to the age at onset, which means the larger the allele, the earlier the onset. But you can see that for a given CAG repeat, this is not really true. If you take 41, it ranges from 15 to 80 again. And the normal allele is not playing a role. But this scheme has to be shown to the people who are coming to be tested, because I think this is an important question coming up all the time. Do I know the number of my CAG repeat? Do I know to tell all the family members? The other thing is that when we have the chance to look at the Enroll database, where you have the distribution of all the sizes of CAG repeats through the people who participated, you notice immediately that it's not symmetrical, and for the small alleles, there is a breakdown. And the question for us was, together with Anne Heinzmann and Sabrina Saya from my team, that we thought, my hypothesis was they are cognitively less affected, the penetrance is reduced, and the disease is cognitively, that's why they're not there. And we showed that it's not really true, because they have a similar cognitive profile if it's a 36, 38, 39, or 40, but they have no chorea. And probably these people are there, but they're escaping the neurologist, they're escaping the Enroll database in a way, because they are not diagnosed as Huntington's disease. Sometimes they're the first person in the family, no family history, small repeats, and no chorea. So I think we should be aware of that. And the last thing I wanted to say about this, because we're discussing this also in the clinic, about the size, again, of the CAG repeat, because it's so important, it's a major player. And you can see here that the reference allele with the CAG repeat is followed by a CA, which is also encoding on polyglutamine and again a CAG. And if you lose this interruption by a CAA, by a mutation gives you a CAG, then the age at onset is earlier. Meaning that the things are happening on the CAG basis and not on the polyglutamine basis. I think that's also an important message to be discussed. And the last thing is the instability. And we had to trace the somatic instability during life. We had the opportunity to take brains from adults that were diseased for Huntington's disease, fetal brains from prenatal diagnosis, and you can see immediately that the postmortem brains are very unstable, very somatic and stable, but not at all the fetus. This is counterintuitive, because in a fetus you have enormously of cell division. In your neuron you have none, and you have a lot of instability in the neuron and nothing in the fetus. First observation. And the second observation is during lifetime, if you look at the age of sampling over 20 years, which is possible now because we follow people for a long time, you can see that the instability is driven by the size of the CAG repeat. Again, back to the CAG repeat. So my last question I wanted to discuss with you and try to answer to people who ask me what is happening before, why we are in a situation where we are born with the mutation, nothing is really happening until 40, 50, 60, 70 sometimes. What is happening in the silent phase before the disease? And so this question is really something that we are, because when you tell someone he's a carrier, in a way, when he asks for the pre-somatic testing at age 35, let's say, this is the mean of what we see usually, I tell them, but you are carriers since ever, so it's just something we look now, but we could have seen it before or we could have seen it later. So the question is, what is happening here before the disease starts? And there are a lot of people who worked on the pre-symptomatic changes, and starting with all of us in Track HD, led by Sarah Tabrizi, where we saw that the atrophy of the striatum, but also of the cortex, starts 7-10 years before the age at onset. We know that neurofilament light is happening in pre-symptomatics before they start the disease, many years before. We know that some impairment on punishment-based learning is impaired very early before anything else is seen. And yesterday we had a nice session on social cognition, which is also something that is happening many, many years before. And Pagnopoulos, who showed that the brains to start with in children have not the same size, because you have in red here the gene expansion carriers and in green the non-expansion carriers, and you see that already in children you have a difference. And we showed with Sandrine Imbert that in fetuses from prenatal diagnosis and in mice, you have an abnormal HD, you have an abnormal neurodevelopmental signature that you can see here, because you have already Huntington in the control, and in the fetus with Huntington's disease is completely differently mislocalized in the ventricular zone. And I think this is something we were really astonished, saying to us that something with compensation have to happen afterwards. And the last thing I want to say, because it's perhaps a possibility to target for treatment, is the fact that in a mouse model, and this again is Sandrine who showed this, in a mouse model, she was able to see that there was a transient synaptic activity that disappeared with high excitability, with dendritic abnormalities that self-resolved after 3 weeks. So this is a transient effect in the mice model, which is not happening in the wild type. If you help this synaptic deficiency for 7 days of short treatment, in this case with ampakine, the adult mouse is not doing the disease anymore. And this should tell us that somehow, to be able to go into the pre-symptomatic phase, to understand the event of cascades that are happening before the disease starts, will give us perhaps the right time to treat with the right molecule and the right time or duration of treatment. So I want to finish here to say that the strength is, as Anne-Elisabeth said, together with all of us, with the families, very important. That's why the meetings, the EHDN meetings are so special, I think, in a way. We have to be around or with the family, it's not even around, and you can see that in this team there are psychologists, neurologists, geneticists, neuropsychologists, orthophonists, you name it. And we are very much linked to the fundamental research, and I would like to thank many thanks to my team at the Paris Brain Institute. Thank you very much.
Thank you so very much, Alexandra. This was fantastic. And now I have the privilege to invite Professor Erich Wanker up to the podium. Erich, he's a professor at the Max Delbruck Center for Molecular Medicine in Berlin, and he's been doing pioneering work throughout the years, really trying to understand the protein in Huntington's disease, Huntington, and what it does and what it interacts with. And we're going to hear some really interesting things from his experience in the laboratory.
Hello, everybody. First of all, I want to thank the organizers, specifically OSA, for inviting me and giving the opportunity to speak at this exciting meeting. As you have heard, I'm a protein biochemists. We work on the Huntington protein, and we are specifically interested to understand what Huntington and different Huntington protein variants do in Huntington's disease pathogenesis. What we know for more than 30 years is that Huntington's disease is a CAG repeat expansion disease. And you have heard about this already from my previous speakers, and this means that there is a CAG repeat sequence in the Huntington gene that gets expanded, and because this sequence is located in the coding region in exon 1, it leads to the production of a protein that is called Huntingtin, which has an expanded polyglutamine sequence. And what we know already also for 30 years is that most patients have CAG repeats in the range between 40 and 55, whereas healthy people have CAG repeats between 15 to 25. And that means that also potentially polyglutamine sequences in the Huntington protein in the range of 40 to 55 are driving or potentially important for the pathogenesis. But what you have to know is that these measurements of CAG repeat size are normally made in blood. And this means that potentially in other tissues in patient brains, you might have different CAG repeat sizes. And actually, this is what has been found out very recently in a very, very exciting paper from Ned Heinz's group from New York University, who showed that in the neurons of patients that degenerate, specifically the medium spiny neurons, you have much, much longer CAG repeat sizes, up to 80 to 200. And this suggests also that potentially very long pathogenic polyglutamine tracts drive pathogenesis. And this is what I would call a paradigm shift in what we currently think about the pathogenesis of Huntington's disease. What I have shown you in my first slide about the disease model or about the pathogenesis actually is very, very simplistic. And the reality, and on a molecular level, it's much, much more complex. And I want to show you a bit on this slide what this means. So we have the gene, which is very complex. It consists of exons and introns that has to get transcribed, which leads to a pre-mRNA, which is very long, which then gets spliced. You get what we call a mature RNA. This actually gets then transported out of the nucleus, comes to a ribosome, and then synthesizes the protein that I have introduced you before. So a very, very long protein, the Huntington protein. But this protein is not alone. It gets cleaved, so you have multiple Huntington fragments that are formed, which then also have a high tendency to form protein aggregates. So you have a whole a range of different molecules that play a role in the disease. But this is not the full story, because the Bates lab has discovered another RNA species, which is called the exon 1 protein. And here you can see this. This is a small fragment that gets formed by missplicing. It also gets produces a protein which is called the exon 1 protein, and also this protein has a high tendency to form aggregates. So overall, on the molecular level, Huntington's disease is highly complex. You have a lot of different RNA species, but also protein species. You have aggregates, different kinds, different types of aggregates that all might contribute to the pathogenesis. Some of them might be more important than others. And to make things a bit more simple for us, our focus in the lab is on the proteins, and specifically on the Huntington exon 1 protein, as well as on the full-length Huntington protein and the formation of protein aggregates. So, having said that, the question that we work on in the lab now since more than 10 years is a very simple one. Are polyglutamine-containing Huntington protein species neurotoxic? And if they are, how do they cause neurodegeneration in patient brains? And to address this, we focus on specifically on 3 protein species: the full-length Huntington protein that you can see here, where the polyglutamine sequence is exposed. It's a very rigid molecule. The Huntington exon 1 protein, which is very small, I have introduced to you from earlier, and the Huntington fibrils or aggregates that we have discovered several years ago that are very rapidly formed spontaneously from this Huntington exon 1 fragment in vitro, but also in patient brains. So, in order to address or to find out what causes toxicity, we work in the lab with a model that is the Drosophila melanogaster. So you can now ask, why flies? Why not mice? The reason is flies actually are very, very clever animals. They have a highly developed brain. For example, a female brain contains more than 100,000 neurons and 50 million synapses. These genes in this model are very, very conserved. And one reason why I like flies a lot is that you can do genetic experiments with hundreds of flies. And if you look at phenotypes or behavioral phenotypes, you can study hundreds of flies and you will get really data that are statistically significant. So, in order to use Drosophila, we have generated a whole set of fly strains, flies that express the full-length human Huntington protein with short polyglutamines or very long polyglutamines that are in the pathogenic range. We have also generated fly strains that could express the exon 1 protein, this little fragment, without polyglutamines or with short or long polyglutamines. And we use a specific expression system or a driver system that allows us to produce these proteins under very defined conditions, specifically in neurons, so exclusively in neurons of this fly model. And what we see is, in my view, quite striking. Here we look at the survival of flies, whereas normal flies survive about 80 to 90 days. This is also the controls, and we see that when we express the full-length protein, only the protein with a very, very long polyglutamine sequence of 145 causes a shortening of the lifespan. The other protein, which is also pathogenic already with 73, has more or less no effect. So it seems to us that very, very long polyglutamine sequences are necessary to cause toxicity in flies. The situation is similar when we look at the exon 1 proteins. Here again, the protein with the very long polyglutamine sequence, 97, is highly toxic, even more toxic than the full-length protein. Not much of toxicity is seen with the other protein, even if you have an expanded polyglutamine sequence. This is interesting, I think, but when we now look at eyes, so we use a different driver that allows us to express the transgene or the proteins in the photoreceptors of these flies. And here we decided to look at very early young flies, so they are 3 days old. And you can see the photoreceptors here with the mutant proteins are still very, very normal. Nothing happens. So it needs longer time to develop a disease phenotype. But when we look at the Huntington exon 1 protein, you can see that the short polyglutamine proteins have no effect. Eyes look very, very normal. But the protein with the pathogenic sequence, the 93 glutamines, you see massive neurodegeneration. So what this tells us is that indeed the Huntington exon 1 protein with 97 glutamines is highly toxic for flies. So when we now ask about the reason for this, we see that the Huntington protein with 97 glutamines forms aggregates in neurons. And this can be seen here, and you see these aggregates already formed very, very early in the development So in 2 days old flies, you see aggregate formation. You see then later also the formation of aggregates with the 97, with the 49 protein, and you can also see this by looking at immunohistochemistry. So what we see is a strong correlation between the formation of insoluble aggregates in the brain and the mortality, suggesting that this is an important process in the disease. So what we want to know is also, can we see a motor phenotype in this fly model? And Huntington's disease is a movement disorder, and so we use the system where we express the Huntington, the pathogenic Huntington proteins in adult flies. So when they are 3 days old, we can turn on the production of the pathogenic protein, the Huntington exon 1 with 97. So we turn it on by adding, it's like a switch, by adding a hormone, and you can see this is the curve, the climbing. Flies like to climb up the glass wall very, very efficiently. And so you can measure this, you can quantify this, and you can see flies that have an age up to 40 days are perfectly okay, but then with age, the climbing abilities decrease. It's a bit like with humans. When you get older, climbing up mountains isn't so easy anymore. But when you basically turn on the expression of this mutant protein, you see a massive decline in this movement. And so I show you a little, if it works, a little video now, where you can see the climbing of the Q17, the control flies, and the Q97 flies. And you can see that the Q70 flies do a bit better than the Q90 flies. So this is basically when you look at D29, where the situation for the flies is still pretty good. But what happens 2 days later, when the flies have an age of 31? And I will show you this in this slide. So you please look at these 3 flies here. And what we see is, so 2 days later, you see that the flies have no capability anymore to walk. They roll over. They show what we call a chorea type of phenotype. There is no chance at all for these flies anymore to climb up a wall. So within 2 days, the flies are really sick, and they have a very strong movement phenotype. 1 day or 2 days later, very sadly, all these flies will have been died. So in a way, we can model a movement phenotype in this model, and this phenotype comes just from producing the mutant huntingtin protein in neurons. So there is no other tissue where the protein gets produced. So we thought this is quite interesting, but what is the reason for this phenotype? And in order to look at the mechanism at the molecular level, we isolated Huntington aggregates from the fly brains. We developed a method to purify aggregates and analyze these aggregates with mass spectrometry. And you can see here this precipitation, as we call, works. We can enrich Huntington aggregates from fly brains. In this case, they were 27 days old. And then we do a mass spec analysis, and we see hundreds of proteins that get enriched from these aggregates in the fly brain. And what we see is that there is a high number of synaptic proteins or synaptic vesicle-associated proteins. And this suggests that the aggregates sequester synaptic proteins, synaptic vesicles, and this might lead to this dramatic phenotype in movement I have shown you before. And we did quite a substantial work to characterize this aggregates in this model, for example, in Drosophila larvae. And what we see is indeed that the Huntington aggregates accumulate in synapses or very close to synapses in neuromuscular junctions. So these are neurons that are binding or that are driving, controlling the muscle activity in these flies. And you see here in green, this is the Huntington aggregates, and the red dots are a marker for synapses. And when you look at the control, the Q17, there is almost no protein there. There's a little bit of Huntington protein there, but not much. So there's a massive difference. And this is before flies are born. This is when in the larvae stage, basically very, very early in development. You've heard that there are already changes at this stage. So we have a model that says that there is Huntington aggregation in neurons, and this Huntington aggregation with the pathogenic protein leads to an enrichment or a clustering of synaptic vesicles very close to the synaptic cleft, and this impairs neurotransmission, potentially reduces the lifespan of these flies. So we have a synaptic phenotype, we have a synaptic disease. And we have actually new data to show that this idea of impairment of neurotransmission indeed influences the survival of flies. And some of the clinicians in this room will recognize this protein, VMAT. It's the target of benzotetrazine, the drug that is used for the treatment of movement in Huntington's disease. So when we reduce the abundance of this protein, we see an acceleration of the mortality in the Huntington's disease flies. So in a way, if we reduce the neurotransmission in the synapses in Huntington's disease flies, we reduce the lifespan of this significantly, the lifespan of these animals. So in my view, specifically at the late stage of disease, we have to take care that synaptic transmission works well. So I summarize our results. So what I wanted to tell you is that the pathogenesis in Huntington's disease is really complex. There are many, many different Huntington protein species, aggregates, but also soluble proteins. Our data indicate that very, very long polyglutamine sequences are important for this phenotype, and we have a strong evidence that aggregation and neurotoxicity are linked. So we think that treating Huntington or reducing the abundance of Huntington aggregates in neurons might be an interesting or an important therapeutic strategy. Just to finish up, this is my most important slide. A lot of people have worked on this. We had funding from different resources, and I want to thank you for your attention.
Thank you so much, Erich. So I would really like to thank everyone, and Elizabeth, Alexandra, Erich, for sharing so many interesting experiences about the challenges posed by Huntington's disease at home, in the clinic, and in the laboratory. And more importantly, for sharing their thoughts about the way families, healthcare professionals, clinicians, and researchers can really work together to overcome these challenges. So thank you once again to all the speakers for inspiring us, and also thank you to the record number of attendees that traveled all the way to Strasbourg to show that the HD community is really stronger together. So thank you.